All right, welcome everyone. We're very excited to see how many people are interested in talking about European Foulbrood tonight. I just want to take a moment to introduce ourselves. So I'm Meghan Milbrath. I'm an assistant professor at Michigan State University, and I'm part research, part extension, and a little bit of teaching, and all of that teaching is in the veterinary school. I've been at MSU since 2015 in various capacities, actually a little bit earlier for a few years too. So I did a postdoc there. I worked as a academic specialist, and now I'm an assistant professor. And I also am a beekeeper, though. I have kept bees most of my life. I've run a beekeeping operation in Jackson County, Michigan, which is South Central, where I focus on queen rearing. And so I wear both the beekeeper hat and the researcher hat for this talk. And then Peter, do you want to go ahead
Yeah.
and
So my name is Peter Fowler. I am a veterinarian. I graduated from MSU in 2020 and have been in their PhD program since. I've been working with Dr. Meghan Milbrath for the last 3 years. My main interest is kind of disease ecology, epidemiology, and my main focus is European foul brood. So I'm excited to talk to you all tonight. So hopefully most of you are familiar with this disease. It's a bacterial disease. It's caused by a super unique bacteria called Melissococcus plutonius. So this is a photo of the bacteria, and we'll see some more in a second here. Basically, what it does, it gets into the larva. What you want to see is healthy larvae kind of down on the lower left. As this bacteria takes over, you'll start to see these larvae get discolored, and then they'll kind of melt into these various stages of goo. This disease in general, European foul brood, has been enigmatic to scientists since it was first discovered. So this is the most recent research. This is done by a group in Japan who's kind of made the most recent breakthrough, which I'll talk about tonight, on studying this disease. And this is open access. So folks want to really geek out on European foul brood. And the first part's a little technical, but the discussion is actually pretty accessible and has a lot of good insight into why we see so many different signs when we're talking about this disease. So folks should feel free to grab that. So I'm going to go back to the beginning and give a little bit of a history, like a chronology of what we know about European foul brood and why we know so little right now. So this disease was first formally described back in 1885. This group in the UK, so this is Cheshire and Cheyne, were the two scientists studying this. They'd known about this for a while. Like this goes back to like Socrates had descriptions of foul brood, and it was kind of general at the time. People were like, "Oh, this disease, it seems to affect larvae. It creates a foul smell." No one really knew what caused it, but these people isolated a bacteria they thought was probably the cause, and they called it Bacillus alvei. So this is an illustration of what they were seeing. You can see it's pretty consistent with what we see today with European foul brood. We'll see a bunch of photos of that later. So at the time, it was just foul brood, and likely what they were seeing was two different forms, our American and our European, and they were probably conflating the two. American foul brood is actually identified more formally by a USDA scientist. This is Gershom Franklin White. So this is about 10 years later. He discovered the spores that are responsible for American foul brood. So that's Panibacillus larvae, which is different than the alvei I just talked about. So this Panibacillus larvae, he was able to isolate it. And then when he gave it to hives, he was able to cause the disease again. So he kind of confirmed this is causing this very unique disease, American foul brood, but it seemed to differ a lot from what they were seeing in Europe. So he wasn't 100% sure that it was the same disease. So that's why I got the name American foul brood. And he called the other one that was more cryptic and they weren't really sure what caused it, European foul brood. So he continued to actually study this disease. This is a photo of his research apiary in New York. And folks were seeing really, really sick larvae. So those are the ones that they would collect, and they would look at those under the microscope and see if they could see a causative bacteria. What he started doing was looking at larvae that were just kind of sick. So before they got the really, really bad kind of melted appearance. And what he ended up seeing in those larvae was a large amount of these bacteria. And he determined that these strange bacteria were probably the cause. And when he fed some of these larvae that had these, the larvae before they died, he would grind them up and then feed them to colonies. And when he did that, he found that they caused disease pretty predictably. So he was like, okay, this is likely what's going on. But when you look at the larvae when they're really, really decayed, you see other bacteria start to come in and take over. And this bacteria tends to kind of sink into the background. Or when he first identified this bacteria, he wasn't able to actually grow it in the lab. So they weren't actually able to purify it and then make sure that this was the causative agent. So for the next decade, or for the next 4 decades, people were trying to grow this bacteria using a mixture of different cocktails. And it wasn't until 56 that we were able to actually find a very specific recipe that we're actually still using today that this bacteria will grow in. So this is some of the first photographs of the purified bacteria after they had isolated it. So they isolated it. Maybe they could study it, make a little bit of progress. But they found that after they isolated it and they cultured it, when they fed it back to larvae, it didn't always cause disease. So this was another big question. Why is it losing its ability to cause disease after this? And that takes us to today. So these are like the big things that happened. They found the bacteria, they were able to grow it, it didn't cause disease. Now we're looking at the genetics of the bacteria. So this kind of started in 2012. So 100 years after the bacteria was first described. And it started in Japan. So that paper I showed you before, Takamatsu, is a veterinarian who is studying this disease over there. And they were noticing in their colonies that there were forms of the disease that seemed to be lasting longer. So one of the typical characteristics with European Foulbrood, and we'll see this when we talk a little bit about the research, is that it tends to appear early in the season. It kind of gets to a really high rate around June. And then when there's a strong honey flow, it seems to kind of taper off and resolve. And it's not uncommon for us to go out, we find a really bad hive. 2 weeks later, we come back and there's virtually no signs at all. And this is without treatment. So this was kind of what was expected in most cases. And they were seeing that wasn't the case in some of these. So they started looking at the genetics and found that there's these 2 kind of genetic clusters. And the one that behaved unusually and tended to last longer in the season, they referred to as atypical. So that's this red one here. And then the ones that people had traditionally described that tended to resolve on their own, they called those typical strains. And why they think that they were losing their ability to cause virulence, and why some colonies seem to have the bacteria and not have any signs, has to do with something called mobile genetic elements. So some of you have probably heard of plasmids before. These are little chunks of DNA that bacteria can get from the environment, or they can trade them from other bacteria. And it's extra from their normal DNA they need to survive. So the green stuff you see here, that's their kind of normal DNA. Without that, they die. These plasmids that they can acquire from other bacteria, the environment can help them survive in certain conditions. Some of these plasmids have some toxin-related genes. And when they culture those, the bacteria, it's kind of nutrient strapped. So what it does is it only replicates what it needs to survive. So it tends to shed those plasmids. And so after culture, you'll see it just has the kind of basic genes it needs to survive, and it loses its ability to cause virulence. So on these plasmids, often you'll find things like virulence, antibiotic resistance that helps the bacteria pump antibiotics out of the cell will be located on those a lot of times. With the typical strains, that's kind of their explanation of why it was so variable disease-wise. With their atypical strains, however, when they repeated these cultures, they still cause disease. So now they can grow these bacteria, at least these atypical strains, and predictably cause disease. And so that's kind of where we're at now. We can actually create this disease in the lab and study it. So, infection versus disease. Do you want to talk a little bit about this, Megan?
Yeah, so that was excellent background, and I think does really speak to why we know so little, because it is so frustrating to work with the bacteria. Another reason why it's very, very frustrating to work from my side, from coming from an epidemiological perspective, is because there's a very high difference between the infection and the disease. So when we use the term infection, we mean like the presence of the bacteria. And when we use disease, we mean when you're actually seeing signs of, damage or issues with the animal. So both bacterial diseases, in fact, have really high levels of inapparent infection. And this is something that's been published. You can see quite a few articles in the lower left, but it's also something we've found in our own research as well. When you have, so this is Giles Budge's work, and you have a yard and you have a colony that has signs of disease, that it's likely that you're going to find the bacteria in all of the colonies in the yard. And we'll talk about this again when we talk about the treatment recommendations, but that's something that makes it very frustrating to study, because there's this huge difference between the colonies that have the bacteria. It could be like all of them in this yard, but then only some of them will be showing signs of disease. So one of the big questions that we have is, why do some colonies show disease and other colonies not show disease? And so when I came to Michigan, that was the big question that I was presented with a lot of commercial beekeepers, especially in springtime and often following blueberry pollination. So at that time where we are in Michigan, the honeybee colonies have access to lots of good food. We had hundreds of colonies. We put them out on fields, and they were beautiful in the beginning. So they were on averaging like 15 frames of brood. And then we would watch them over a few months, and then we would look to see how many got sick. And some of them would, get so sick that they would die or dwindle down to nothing. And some of them would be perfectly healthy in the same yard, which that's something that's really frustrating and difficult to understand. When you see that type of variability, we look at the different factors. So in the gray box, you can see some of the things we've been looking at. And I can tell you, for those of you who want to cut your night early, I can tell you that we have no answers. So as an extension, the extension half is incredibly frustrated. My science researcher half likes the job security, I guess. But what we've been looking at, you can look at the host. So I'll talk a little bit at what we've looked at with hygienic behavior, but there may be differences in the bees with different immunity. They may have different genetics. We are looking a lot, or Peter's looking a lot, at the pathogen. So he mentioned these toxic plasmids that they have. He'll talk a lot about his work looking at strain type. One of the things we're not talking about tonight is we've looked at co-infection. So we looked at deformed wing virus and just whether or not the pathogen load is much higher. And then the environment. And this gets talked about a lot with stress. We've done some research, which we won't be talking about tonight, on pesticides. We've looked into nutrition. It could be density. It could be competition. There's all these different factors that could affect why we see it show up in some colonies and not in others. And I'll talk about some of the research tonight. All right. So it is very sparse. My goal is to try to talk to as many people as possible and get more people to do EFB research, but we've been working on it since about 2018. And so what we did the first few years is do what's called a prospective cohort study, where basically you get your cohort of hives that you're looking at, and then you follow them throughout the summer. So we enrolled colonies that were all healthy. So they had to look good at the beginning of the year. They had to be queen right. And then we watched how many got signs of disease, and we recorded a lot of data about them. The big message here is that we found about 50% of these colonies were developing disease, which is horrifying. I'd heard it was bad from the beekeepers before, and it wasn't that I didn't believe them, but it's hard to think about a disease being that bad and it not, people aren't shouting it from the rooftops, and it's not getting tons of government funding, and there aren't more programs. One of the questions in the chat says, why are we seeing more EFB and honeybees coming out of pollination and blueberries both in Canada and Maine. And I'll add to that list also, I've heard it from New Jersey and Quebec and British Columbia and Oregon and Michigan. And one of the things that we have found is, I got started on this. You can see these are hives in blueberry fields. I guess if you know what blueberry fields look like. And so I got started on this because of this question about it related to blueberries. However, we also find it quite often in colonies that are nowhere near blueberries and have no association with blueberries. Peter will talk about the seasonality, and the seasonality is very closely timed to blueberry bloom. And while there are a lot of colonies that come out of blueberry bloom and develop disease, it's not necessary in order for them to get it. So when we started our trials, we looked at them basically right when they came into Michigan, which would be about mid-May. And then we look at them right when they leave the blueberry field, which is usually beginning of June, and then a couple weeks later, which is like mid-June. And then some of them we followed all the way into September. And as Peter mentioned, we used to think about EFB being this like spring brood disease that just clears up. And we definitely had colonies that were showing signs of active infection all the way through September. And this is in the context, even when mites were controlled, that we would see this. All right, so one of the things we did look at was hygienic behavior. I'd heard from lots of people that there was no association with hygienic behavior, but honestly, there were no scientific things published on it. In 2020, we followed 30 colonies that were on blueberries and 30 colonies that were not on blueberries. This is a project funded by Project APSM. And we found basically equal rates of European Foulbrood disease in them. And then we did hygienic testing twice on all of them. And we did have some of the colonies that had very, very high, were very nice hygienic. Like you can see this one on the right. Well, this is a freeze kill assay, so we can tell it's highly hygienic because they removed all of the ones and then they kept removing more. But there was no association. That's basically what people thought, but it hadn't been shown. So the hygienic behavior, especially this freeze kill assay type, has been shown to work for chalk brood and American Foulbrood, but not for European Foulbrood. It's still a nice trait to select for those reasons, but it also is associated with lower mite risk. So it is still a nice trait. It's just not going to solve all of your EFB woes. Other thing that we looked at was protein supplementation. So a lot of times people talk about, European Foulbrood is caused because blueberry pollen is so low in protein and, or there's not enough food. And so what we did is we followed 14 different blueberry yards, and we followed 9 colonies per yard, and they were all enrolled in different treatment groups. And so some of them, their treatment was to get protein patties. So the idea is that if there's a protein limitation coming in the spring, that's causing stress for the colony, and then it's causing them to have higher rates of disease, or especially if blueberries aren't good food. The two things that we found is, one is that the protein patties did nothing, which is a shame because that would have been just a lovely and elegant solution. Just throw protein patties in there and everything's solved. So we did not see any benefit to putting protein patties at that time. And for us in Michigan, it totally makes sense because we were also pollen trapping from those yards to look at incoming pesticides. And we were getting just gallon bags of very diverse pollen. So we have published the paper on what pollens were coming in, and it's many, many different species. We're currently analyzing those pollens to look at the protein lipid ratios. But when you have large amounts of diverse pollen coming in, that's a pretty good sign that they're not protein limited. The colonies also didn't act like they were protein limited. So we did see signs of disease, but on blueberries in Michigan, they also have a lot of nectar coming in, and we had colonies swarm. We had, they're building honey and they're building up the ones that aren't sick. So at least in our region and the way that high bush blueberries are grown in Michigan, the European Foulbrood is not related to a food deficiency. So we also have been looking at recovery options. So we looked at giving them protein as a treatment as well. That also wasn't effective. We combined it with antibiotics, like you can see here. We've looked at prevention with different timing of antibiotics, and we've looked for both prevention and treatment with probiotics. And the antibiotics in our cases were still very effective. They did cause disease to clear up faster than the probiotics and the protein, but we don't have a great recommendation. Like, if you do this, you'll never have EFB again. We were really hoping that we'd have these different timings and different things that we could test, and we'd have a good recommendation. And we're not there at this point yet.
All right, so I'm going to talk a little bit about my project. When I first started, like Megan said, we had some data about certain operations and certain yards that have been dealing with European Foulbrood, and we knew the rates were high in some areas, but we didn't know how widespread this problem was. So what we did was this kind of broad 2-year surveillance project, and we included a number of different commercial beekeepers, sideliners, and hobbyists. And what you see in gray here, these are all the different counties that were included in this surveillance, or I would say represented. This was close to 1,000 colonies. We were looking at signs of disease, but then we were also taking samples of the larvae and actually looking for the bacteria. And what we found was overall, and this was the case across different operation types and sizes, about 1 in 3 colonies had this bacteria present at some level. And a lot of them had both strains, typical and atypical, present. So this was pretty surprising to us. And then, like Megan said, that there's a strong seasonality to this. So this is the percentage of the colonies that we screened across all different operations. That line represents the percentage in each month that were positive for this pathogen. And you can see in June, over 60% of the colonies had Melissococcus plutonius present. So super high rate, and it doesn't really get below 20% at any point. And this tracks pretty well with what we were seeing clinical disease-wise as well. So this is the percent of colonies that were actually showing signs when we took the samples. And you can see this kind of gap between this red and blue line. These are the colonies that had the bacteria present but didn't have any clinical signs. So you can see there's a pretty big portion, almost 20% of them. So the disease was everywhere. The pathogen was everywhere. The next question we had was, like, why is it persisting? And so this is Robin. She had this project a couple of years ago as part of the College of Veterinary Medicine summer research program. So she was working with us. Her main question was, how long does this bacteria survive on different surfaces we come across in the bee yard? So there's a lot of data on American Foulbrood. We know those spores like last forever, right? But no one's really looked at Melissococcus plutonius. I think people have assumed, because it doesn't form a spore, that it's probably like it dies fast in the environment, but it hasn't really been specifically looked at. So what she did was she took a number of different sort of materials. So we had wax, wood, washers, cloth, honey, which was a little tough to work with, and gave us some mixed results. The vial there is the honey. These bags are the different samples. And what she did was inoculate these with a known concentration of the bacteria. We did this for atypical and the typical ATCC, a reference strain people use in research. So we had representatives from each group to see what the differences were and how long they survived on these materials. Basically, she would set these up and then have replicates. So she would pull one of these samples and then culture it to see if the bacteria was still alive at each of these time points. And at 2 weeks, both of them were surviving. So you can see at the top, these little dots here are the Melissococcus plutonius growing after those 1 to 2 weeks. And then she set up a couple of samples for us to retest long-term. So she set up a 1-year sample. This is the differences between types. So the atypical strains tended to survive longer. The typical ones were kind of variable. Sometimes they survived, sometimes they didn't. But the atypical remained viable on all the surfaces when we recultured it 1 year later. So something's going on with this atypical strain that's causing it to be very persistent in the environment. And that's kind of like leading into our next phase of study. We're also interested in treatment. So people have been using oxytetracycline on this disease in the United States since like the 50s. And like we know with a lot of other pathogens, resistance is a big concern, right? Most strains are still susceptible. There's only been a few studies done actually seeing if Mosacoccus of all these strains is still affected by this antibiotic. Some of them have found mild resistance. It doesn't mean that treatment isn't working out in the field. This is kind of what they're doing when they say resistance. So going vertically, you have different concentrations of the antibiotic. And then you basically are putting bacteria in each of these vertically and seeing what concentration it stops growing. They found usually Mosacoccus stops growing in the first 2 wells at the first 2 concentrations. They found it 1 higher. So this may be important, but it may mean nothing for how we're treating things at the colony level. So it's been found in the lab, and the strains that they have found that showed this characteristic it's been like 3 out of 70 or so that they've tested. So when they do find it, it's fairly rare. We've done some preliminary studies. So this is actually one of our plates that we did here. And the column you see on the right there, that's a bacteria, that's a different bacteria that is resistant. And you can see how it's growing up the whole column. The one on the left here is Mosacoccus. And the strains we've tested so far have been pretty susceptible. You can see it's not even growing at that first concentration there. Good news so far, this is something that's ongoing. So we're continuing to screen for resistance. We have around 300 different isolates of this bacteria from all these different colonies. So we're going to be screening all those. Hopefully we'll have more data on that coming up. But the next questions we had was what actually kills this bacteria? So we'll be, now that we have all these different strains isolated, we can put it on different material, and then we can see, how do these different commonly available disinfectants kill it, and how much survives after we disinfect. And then we're also going to be looking at the properties of propolis and its ability to keep the disease itself at bay. So these are some of the things that are kind of in the pipeline, and hopefully we'll have a lot more information on this stuff probably this time next year, I'd imagine. All right, so now we'll go into diagnosing. On the right, you see what you typically expect from European Foulbrood. And I like putting it next to this illustration from like 1885 that like looks basically exactly the same. We're still dealing with this all this time later, and we still have a very basic understanding of what's going on. So the first thing you see is like spotty brood. You pull your brood frame out. What you expect to see, you see on the left here, nice clean brood, pearly white larva. You see a spotty brood pattern. This could be a number of things. EFB, AFB, both those can cause this. But then we've also seen this with high mite loads, chalk brood, high virus loads. So other things can cause similar signs. So when you see something like this, the first thing obviously you want to rule out is American Foulbrood. So I won't go too into depth with AFB testing. There's a lot more available for testing American Foulbrood. And a lot of the same tests that you would use for European Foulbrood, like sending it to the USDA or using this rapid test, is available for European Foulbrood and AFB. Mostly what you're doing with this is trying to make sure you don't have American Foulbrood. So signs that are consistent with this, they say generally this affects older capped larvae. So a lot of times you'll see these perforations of the capping where nurse bees have kind of peeked underneath to see what's going on. They think the larva's dead. European Foulbrood generally, not always, generally tends to affect brood at a younger stage. So when you see a lot of older brood sick like this, you kind of lead towards AFB and you want to do some testing to rule it out. You'll have, of course, sunken cells that are kind of, they can be kind of greasy looking. And then when you do a whole milk test, which we'll show you what it looks like with EFB in a second here, but when you do, or not whole milk test, the matchstick test, and that's where you stick a stick in there. And if it's American Foulbrood, it forms this caramely, sticky, ropey substance. And as you pull it out, it'll form a long string. And with European Foulbrood, it tends to be more watery. And you'll see that in a second here.
And I just want to jump in for a second on the perforated cappings, though. So if you go back to that original drawing, one of the things we noted was that there's actually a lot of perforated cappings there. Mm-hmm. And a lot of people mistakenly think that if they see perforated cappings, then it means they say like, oh, AFB is cappings, EFB is open. AFB is cappings in older larvae, but it doesn't include the younger larvae. EFB can have the cappings, but it's also younger larvae too. So that's something that we do see kind of misprinted or out there a lot, is saying if it's, you know, these sunken cappings with the holes, then it has to be AFB, and then people get all panicked. But you get both, the scale is a little different, and like the holes in the cappings you can get with both diseases. Thanks, Peter.
Yeah, it makes sense that there's all these various different signs too, when you think about both those different strains being in the hive, and with all the different characteristics that both those strains of Melissococcus have, they're killing it at different times. And so you're going to see a pretty wide variety of signs with European Foulbrood. So you want to rule out this. This is something we see really commonly in the fall. Hives can look a lot like European Foulbrood. It can be a lot of open brood that's very sick and kind of starting to melt. It's very discolored. But usually this is associated, especially later in the season. You saw that that graph I showed you where European Foulbrood tends to go down, not always, like Megan said, like we see severe cases later in the year too. But as a generality, like it's less common. And when we have really high mite loads, though, you know, whether it's the mites themselves, like you can see mites on some of these larvae, whether it's the mites themselves or the viruses they carry, it presents very similar to European Foulbrood. So we see that in September, October-ish, and it's usually accompanied by high mite loads. When we see this, like looking at some of these frames, I'm like, that's probably European Foulbrood, and it can have both. But we've seen these signs and tested them and not found Melissococcus plutonius. So there are other things that can look like it. So just be kind of aware of that. So symptom-wise, this is kind of what you expect for classic European Foulbrood. So up on the left corner, you see like nice, healthy, pearly white larva. It's plump. As the bacteria starts to take over its midgut, it'll start to kind of discolor into this yellowish. And one of the characteristics that you see is, I don't know if you can appreciate on the photo, these white lines on this photo here. I don't know if you can see my cursor. I get my laser pointer out. Okay. So these white lines here are actually the larva's tracheal system. So as the disease progresses, that tracheal system becomes pretty obvious. And that's one of the classic signs that you see. And then the larva starts to twist in its cell. And like I mentioned back when I was talking about when they first found this disease, as the disease progresses and it gets more melted, the bacteria that causes the disease tends to be harder to find. So when we're doing diagnostic sampling, when we're taking a sample and we're going to send it to USDA, or we're taking a sample and we're going to do another diagnostic test, we're really looking at taking these early samples here before that other bacteria has taken over. And when Melissococcus plutonius is at a higher level, and you're just going to be more likely to catch the bacteria in your sample if you take those samples. So and then obviously, like Megan said, you can get these little off-center cappings as well with European Foulbrood. So this isn't just indicative of American. And then the scales with American Foulbrood, the scales are like black on the bottom. European Foulbrood tend to be more flexible, scaly, or rubbery, and they tend usually to be more towards the back of the cell, and they can be darker, but they're not black and sticky like the scales for American Foulbrood are. So you can see these tracheal systems are also evident in those.
Well, and there are two types of scales so there's this one that we were calling clear scale. So we have names for all of these, because one of our really fun projects that Peter got to participate in is where we got to take pictures and individual samples of 10 yellow blobs, 10 trachea, 10 twisted larvae, 10 white blobs. And so but we had two different types of scales. This one that's shown here, we were calling clear scale, which I understand that it's not clear. Well, even the one to the left, but you could see the trachea through it. The one that's not pictured is where the larva does tend to dry out straight. So the one on the left-hand side of the middle row, you can see the outside ones of the middle row, you can see that they're actually along the bottom of the cell, or what would be the lower wall. They'll dry down to like a sticky black, but they'll be able to be removed. So they'll get as dark and they'll look really similar to an AFB scale for the people that are familiar. But the AFB, if you were to try to remove it with a tweezers, you can't remove it. It's stuck to the cell wall. Whereas sometimes with the EFB, like if you tipped it over, they could actually fall out. And I've seen them on the ground of bottom boards before, almost similar to chalk brood. But it can look pretty similar. And I've had people call me in a panic about that one as well. So, but yeah, I think we'll be able to see it in some of the upcoming slides.
So these are just more slides to just show how variable this can present. To kind of see these discolored ones on the side. Starting to get darker, and you can see, like Megan was saying, this cell here is kind of starting to sink down towards the bottom.
That one's a really good one to show all the visible trachea.
Yeah,
I think that
obvious here.
Yeah, Rob has some really nice pictures about showing the trachea. And colonies they can have many in one frame. American Foulbrood is much more consistent. Like you either see the larva dying and the cappings, but when you start to see yellow and brown and gray and twisted and these trachea and all of these different things happening at once, then that's going to be much more likely to be European Foulbrood. It'll be very, very diverse and very colorful. So when you start to see lots of, an American Foulbrood is really just like caramel color to black. When you start to see those lovely yellows in there, then that's going to indicate that you're probably towards European Foulbrood. And then it does depend, like the reason you see so much diversity is it depends on how old the larvae were when they got infected and when they started to die. So this is the photo I was talking about with the black scale. So in the middle at the bottom, you can see, in those, they dry down and they look really similar to American Foulbrood. And that is just the larva or the prepupa are starting to stretch out. And you can even see one with those jaggedy holes in that scale in the middle there. This would make me think about American Foulbrood, but really what's happening that makes them look similar is that the larvae are dying at that same age. So EFB kills younger larvae as well, but they're also killing these larvae that are right around that, even getting into that pre-pupal state or that pupal state once they're capped.
And a lot of these secondary signs that we're seeing is also due to other bacteria that's just present in the hive all the time and just happens to make its way into the cell when the larva is starting to decompose. So it's kind of creating a lot of different signs as well. So, and that secondary bacteria is also why they think that strong odor comes about too, is Enterococcus faecalis. It's one of those secondary invaders.
Yeah. And a lot of these that are just kind of the little white chunks, like that one that looks, this one looks very suspiciously like AFB and like parasitic mite syndrome, which it's nice to be able to rule both those out. Yeah. And so part of the way that you do your diagnostic is by ruling out those two other things.
And keep in mind, it can have both AFB and EFB. In fact, when you're thinking about like how prevalent EFB is here, like I would kind of be shocked if a hive had AFB and it didn't have EFB, but
and we've definitely, so here's one where it looks like you see a little sac brood. You can see another one of those like scales that looks suspiciously like AFB scale. And like these white melted ones are the ones that parasitic mite syndrome will look like. And, one of the things that's hard is when you're looking at these larvae, like these larvae are basically dead. You're not doing a health examination on a live animal. Sometimes you're trying to figure out how it's dying. And so if it dies from a virus or at a bacteria at the similar age, it may look very, very similar. And then we saw a lot of chalk brood as co-infection. I mean, not a ton. We saw normal rates of chalk brood, but it was often present as a co-infection. And sac brood, and as mentioned, we had deformed wing virus and Lake Sinai virus and sac brood virus, and were all present as co-infections.
So this is the lateral flow device I was talking about. It's basically a rapid test like the COVID tests we've all been taking. So with this, I like to stress the samples you select are really important. If you select those older larvae, you're going to be less likely to have the bacteria that causes the disease. So you really want to select larvae. If you're curious about European Foulbrood, you want to select the larvae that are still kind of alive but discolored. And I would grab a couple of them for this test. So basically how it works, you throw the larva in this bottle that comes with it. It's got some like buffer and some beads in it, and you shake it up to kind of bust the larva up and expose that bacteria. And then you just take a couple drops and drop it on here. And I think it takes about 15 minutes and you'll get a test positive line. You can see this one was positive here. Just make sure that the test you're using, because they look identical. One's for AFB, one's for EFB. So they're specific to that disease. This one here you can see is AFB. So, and this is one of the tests for European Foulbrood, at least. We have done some validation. So we wanted to see how accurate this test was when compared with some of the other testing. So kind of the gold standard is sending it to the USDA and having them test. So we worked with the USDA on this project. We submitted a bunch of samples that had European Foulbrood, and they used the test and they compared it to their test, and they found that it was fairly accurate. There's possibilities of false negatives, but if you get a positive, it's going to be positive for that disease. So super useful test to have out in the field. And then this is the matchstick test we were talking about. Is this in the video? Is it? No.
I think it is a video, but it might not.
Okay.
So one of the descriptors that we have been using that seems useful is that EFB is boogers and AFB is snot. So it's still, this one we've tried to get it out, but this one's pretty dry. I think the next photo will look that it's still kind of, it's still kind of goopy, but it really isn't going to like string out and snap back.
Yeah, it's a lot more watery. Sometimes it will string out a little bit, but usually if you look close at it, it's kind of like the remnants of the different organs of the larva that are pulling out with it. So yeah, kind of like that, as opposed to, I think there's one. This is what you'll see with American. It's very, very sticky.
And so this is something that you're doing with the older larvae, but I think I was muted, but I was trying to emphasize the point that with those lateral flow devices, when you get those negatives, a lot of times it's people picking these gross old ones to test. So at this point, when you're getting these old goopy things for the matchstick test, there's not going to be any Melissococcus left. It's just going to be secondary bacteria. So if you were to test like this dried goopy one with the lateral flow device, it would likely show up negative. And so that's something that I think is really important, because a lot of people talk like, oh, they don't work that well. But I think a lot of times it's, we did a test with many, many of them, but we were very, very careful about selecting those very early stage larvae.
So this is the send to Beltsville slide. The website to get there is kind of long. So I tell people just to Google Bee Disease Diagnosis Service, USDA, and like this is kind of the first thing that comes up. So their directions haven't really changed in the last 100 years. So this photo is from a USDA bulletin from like 1930s. So they've been doing this for a really long time, have a lot of experience, but don't expect a really fast turnaround. It's a free service, but it's like one guy serving the whole country. But people should make use of it. It's nice to have reliable diagnosis, and then it's also nice for them to have data on their end as well. When you're collecting these samples, they want you to put it in paper or cardboard when you send it. One of the biggest issues they have on their end receiving samples is people put wet stuff in paper bags, and mold can start to grow. And that's
Plastic
kind of
bags.
what did I say? Paper?
Yeah.
Oh, yeah, plastic bags. So no plastic bags. Read the text, don't listen to me. So no plastic bags. Send in something that's breathable. And that'll be a lot easier for Sam, who's the diagnostics guy over there, to read.
And we did ask him about like sending in, because he wants a chunk of comb, which is easy to cut if you have beeswax foundation. It's hard to cut a chunk out if you have plastic foundation and you're sliding it underneath. But he does do a visual assessment as well. So if you can get like a nice patch as much as you can, or not as much as you can, but like a nice big patch, because then it will also give him the option to select some of these younger ones that he can try to see the bacteria off of. So he has multiple ways that he can do the test, but so it's nice that he has the options and he can look at it as well.
Yeah, and he does mention also if you are cutting out a chunk of comb like that, try to avoid the honey or any of the nectar, especially. Like that can also lead to a lot of mold overgrowth in the sample. Don't really worry about squishing the larva or anything, because the bacteria actually persists within the midgut inside that larva for at least 3 years at pretty high rates. So it's usually pretty easy for him to read on his end, as long as the larva is not moldy. So just to drive it home, if you're doing the lateral flow device or sending it, if they're looking for bacteria, if you're sending it to USDA, try to avoid these late stage samples. You really want to be looking at these earlier stages.
Yeah, so now we're getting into the stuff. So there is not a good treatment recommendation for everybody. So there's not going to be a thing that I can say, if you have European Foulbrood, this is what you should do. And a lot of it depends on what is your likely risk of reinfection. So if you are in a place where you're super remote, you only have a few honeybee colonies, and your chance of reinfection is very, very low, it may be absolutely worth your time to treat it like American Foulbrood, like you see here, and really try to eradicate it from your operation. What in talking to people and what we're starting to see with our data is like when people have it in their operation, it's kind of in their operation. And also with a lot of the European data where they've tried to eradicate it, you're able to eradicate it for a while, and then it tends to come back to a particular area. And so you could treat it very seriously and then get rid of it. But if you're in a place where there's lots of bees and beekeepers, which in Michigan, that's basically the whole state, then you're going to really try to aim for control and minimizing disease. So there are lots of options, and we'll go through them quickly, and then we can talk about more when we do the Q&A too. So one really, really important thing in your treatment is that you can have the bacteria present without signs of disease. So a lot of times people will say like, okay, I have this sick colony. How do I deal with this sick colony? Or I'm going to move this colony that has signs of disease into a quarantine yard. And when you're doing that, you're not moving them, just into a quarantine yard. You're not moving all of the disease into the quarantine yard. You're just moving the one with the visible signs. What we see in operations, including ours at MSU, is sometimes you'll have colony A get sick, this year, and next year colony A will look beautiful and perfect in the spring, and colony B will be sick and it had shown no signs of disease the year before. And the next year it's a different one, and you're kind of playing this game of whack-a-mole. So what you want to do is really focus at the yard level. So that you're managing disease at the yard level, because a lot of it is going to be in the adult bees and the nurse bees. One option is watchful waiting. As we said, like a lot of the treatments actually clear up. And for us, it's always when I want to bring students out to see it. And so I'll be really excited and we'll mark this colony. And then by the time the vet school rotation starts or the bee club meets, it'll totally look fantastic. And historically that was, and I think that's the reason it doesn't get a lot of attention, is watchful waiting was what people used to do. Caveat is that you wouldn't want to do this if you had a colony that was so weak that it was likely to get robbed out. So this would be a big, strong colony, plenty of time left in the season, and you could, see what happens. What we tend to do is kind of evaluate disease based on the number of cells where we see signs of illness. So if it's, you go in there and you do an inspection and you see 10 cells, you're like, well, that's not so bad. It's a strong colony. Let's see if it clears up. And the next time you go in and there's 100, then you're probably going to want to, make some changes. I'll tell you some real depressing things that don't work. We got very excited to try to do partial shook swarms. And what I mean by that is we basically removed the entire brood nest. So the idea is that we got a break in the brood cycle and we removed a lot of the pathogen, or that's the thought behind it. So we took out all of the frames that had brood on them, gave them a full break in the brood cycle, shook the rest of the bees in, but left all the frames with food and honey. And we saw infection come back that same season. But we were just trying it. We didn't do it experimentally, but just tried it at a small scale. And on a small scale, it wasn't enough to get rid of disease. One of the issues with European Foulbrood too is that even when it clears up, so let's say you don't see any more signs of disease, you see that spotty brood pattern. And that spotty brood pattern can persist as the queen, goes through and fills in holes, and then they emerge and she's filling in holes. And so that pattern can exist for a really long time. We are looking into this year doing full shook swarms and actually doing this at the yard level. I have a question mark because I don't have a great, do exactly this thing and it will solve all of your EFB problems forever. But the idea behind that, and I'll show some photos of it, is you're getting rid of all of the frames and putting in foundation. You can do this with or without antibiotics. So we've done this a few times for American Foulbrood, both with and without antibiotics. And it works great for that. And then the other thing is you can think about doing this at the yard level. So if you are a beekeeper that has quite a few colonies that are affected by EFB, it doesn't make sense for you to burn all of your equipment every single year. So what you can do is practice in biosecurity, kind of splitting your operation into different sectors. And so you can do a shook swarm in one yard and take all of that equipment that likely has the bacteria present and move it to another yard that has the bacteria present or signs of disease as well. So you're kind of having a clean yard, and then things can come out of that clean yard, but they can't go back into that clean yard. And then the next year you get another clean yard, and the next year you get another clean yard. And, we usually recommend doing 20% or 10 to 20% of your frames are new, but you're doing that always out of those dirty hives for a while. So this is us doing a shook swarm. So the colony on the left that the beekeeper is standing behind is the one that's sick. And the colony on the right is going to be the brand new equipment. So there's frames in the bottom that are just foundation. And then if you want to click through, we just shake all the bees in. So we took the lid. And in this case, this was actually a colony with AFB. We incinerated, we put it in contractor bags and incinerated. We brushed all the bees off. That is my patented fancy bee brush that we used, but that was nice because we could just throw that away as well. And you just shake all the bees onto brand new equipment. And then in that case, we'd feed it. And this is an option that's only available to you, obviously, at the beginning of the season when there's enough time to grow. So here are the steps. You shake them onto foundation. The box on the top, that blue box, was just a funnel. So we were just using that to make all the bees go down below. And then again, you can pair that with antibiotics, but it's not necessary. All right. So another thing that doesn't work, I mentioned these before, we tried just feeding with protein patties. That was not sufficient to make it go away. But in our case, we were definitely never protein limited in Michigan. So that didn't seem to be at least the stressor that caused anything. The other thing, that's just have an asterisk on it. So we have tried probiotics and we don't have a good recommendation. Part of the issue with that, though, is it's not that I don't think probiotics are valuable, especially in theory. There's just a lot of research that needs to be done on which ones and the timing and things like that. So this is something that we've still, are looking into, but we have tried it, like I said, both as a preventative and as a treatment and haven't seen a difference. I do know that a lot of people really like using the probiotics and feel very strongly about them. And I respect everybody's beekeeping as long as their bees are healthy. One of the things that is really useful, though, when you are trying a new treatment, especially an expensive one, is to make sure that you have a control. So everything that we do is we test the treatment, and then we have a whole group of colonies that don't get the treatment. And one of the difficult things about European Foulbrood is that it does often clear up on its own. And that rate of it clearing up on its own, if you have 10 colonies and you just leave them all, some of them are going to get better and some of them are going to remain sick. So you have to kind of try a treatment on a lot of colonies and see if the number that get better are much higher than the number that would get better if you did nothing. And the number that stays sick is going to have to be lower than the number that stays sick if you do nothing. So it is really important when you're evaluating these things to make sure that you do have a control case in there. A lot of people are very interested in essential oils, and I know there have been some beekeepers that have been talking about tea tree oil and eucalyptus oil, and I think someone put a question in the chat about another garlic oil. And Peter will talk a little bit about the research, but I'll just talk a little bit about those studies in general. Is you want to make sure that when you're talking to a beekeeper who's tried stuff, that it's in lots of colonies and that they had a control to compare it to. I mean, at least you get some data of, well, it didn't immediately kill all the colonies right away. But those are the things that you want to ask if you want to evaluate whether or not something worked. And there are beekeepers who say, like, yeah, I'm just trying this out and here's what I found. If you want to just experiment and try stuff out, that's also incredibly valuable. But when you're experimenting with things like this, especially with oils are chemicals, and they can absolutely be very damaging to bees, and they can absolutely build up in the wax, and they may not be healthy to either the larvae or the adults or to humans. So you want to be very, very careful. You want to mark frames. And then I would only use about 10% of your colonies when you're trying something new. The SARE program, which is a USDA program, has a lot of lovely grants that you can get for farmers and ranchers. And they also have a lot of guidelines on how to set up little experiments as well. So I don't know, Peter, if you want to talk more about tea tree oil in specific.
Yeah, there has been one study. Someone asked like any specific studies. There has been one study, and it was co-authored by that guy from Japan, Takamatsu, that I had mentioned is kind of one of the leaders in researching this disease. And what they were doing in that study is they were putting this into nanoparticles as a formulation. And the idea was that these nanoparticles on their own, they're a little bit antimicrobial. But they also can shield some of the toxicity. So what they were concerned about was some of these oils, like Meghan said, these are chemicals, and some of them can be damaging to honeybees. So what they found was that at least when they're testing the bacteria in the lab, which you saw that little plate of broths. So they did something similar to that with some of the chemicals in these tea tree oils. And they found that it is pretty effective at killing the bacteria. So the next question was, if we just use tea tree oil on the colony level, is it going to kill the bees? So what they did was they sprayed some of this tea tree oil on bees, and they watched them, and like 80% of the bees died, like by day 4. So on its own, at least when they're spraying it, like it can be pretty toxic to the bees. When they sprayed the nanoparticle formulation of it, it seemed to be pretty effective. Well, they haven't tested it in the hive level. It seemed to be less effective at killing. So all the bees survived when they used the nanoparticles. I would still be cautious around using oils, like Meghan said. There hasn't been a follow-up study of that I know of yet, but it could be an interesting treatment if they find some other formulation or way to get that in there. So
get your hands on some nanoparticles. I'm just kidding.
Yeah.
A lot of people ask about the vaccine, and there is a commercially available vaccine for American Foulbrood. That same company has been looking at European Foulbrood as well. At the moment, there is not a vaccine that is currently available for European Foulbrood. And also, we're not sure that it would work exactly the same way. So the vaccines don't work the same way as they do in other animals where they cause like an antibody response. They really count on the system of transgenerational immune priming, which isn't the same for all the different bacteria. So, it'd be lovely if one came out, but we're not at the point where it's available yet. And then this one I'm particularly excited about. So is a phage therapy. This is another one where there's more research on the American Foulbrood font than there is for the European Foulbrood font. But I just did talk to somebody last weekend who's interested in looking at this. So I'm very excited. So phages are basically viruses that affect bacteria. And so we can look for something that is naturally found that can affect the bacteria. And a lot of people just go out and screen for them. There are some people that are starting to look to see if they can find some that will kill Nosococcus plutoneus, which would be fantastic. And then the last treatment recommendation we'll talk about is antibiotics. So as Peter mentioned, the oxytetracycline is the only one that's available for control of European Foulbrood. So there are 3 antibiotics that are labeled for use for American Foulbrood. Only one is labeled for the control of European Foulbrood, which is oxytetracycline, which is the fancy name for terramycin, or you may know about it by Tetra B or a lot of the other product like brand names of it. So here you can see we're applying it to a hive. It's often found in this powder form. This is where it's mixed in with powdered sugar already, basically. And you apply it at a set dose that's labeled on the bag, but it's basically a couple tablespoons, and you put it around the brood nest. And then the bees take it in and you apply it 3 different applications, 5 to 7 days apart. But all of these would be on the label that you'd follow or on your instructions. There are also formulations where you can put it into liquid feed or when you can put it into like an extender patty. But this, I think, is probably the most commonly used or most recommended method that we see.
So antibiotics are a little bit harder to come by recently. As many of you probably know, you need a veterinary oversight. So you need a veterinary who's willing to establish a veterinary client-patient relationship with you and is able to write either a prescription or a veterinary feed directive. So a little piece of paper, basically, you turn into Daedant or Manlake or any of these companies that sell these antibiotic formulations, and then they can release the treatment to you. We're working on getting more vets trained to help out with this. Right now, you can go onto the HBVC. So there's the Honeybee Veterinary Consortium. Yeah, if you Google Honeybee Veterinary Consortium, you'll find it. But they have a little map finder, and you can pull up Michigan. Unfortunately, right now, there's only 4 veterinarians listed. And they do also have the veterinary feed directive that the vets need to fill out on that website. So if you want to work with your local vet and point them towards there, they have resources they can give to.
And if you have a vet that you're working with that isn't comfortable taking bee clients, we're in the first year of a 3-year, very well-funded project to train veterinarians. So we're doing hands-on clinics. We've got an online course. We've got a survey where we're, trying to understand what more they need to know before they're comfortable. But one of the things that you can do is just have them call me. And even if they're not in Michigan, I will happily talk veterinarians through what they need to know so that they can prescribe something or write a VFD without losing their license, because that's going to be their big concern. And one of the things that is comforting to them is if they can talk to someone that can tell them, like, this is standard practice, these are where it's labeled, this is how it's commonly used, so that they know that they are using it appropriately. So and then also you can put them in touch with other extension as well. But this should hopefully get 1,000,000 times better in the next few years. All right, so here's the big point. So one of the things that we know is that it persists on equipment for a very long time, which the part that we haven't done yet is to see how infectious it is after that equipment, but we're at the year mark. We have more funding to look into it, but this is actually very interesting to us as scientists because it doesn't form a spore. So it shouldn't be that persistent, but it really is persistent. So you want to be maybe a little more respectful with your equipment. Another main point is that even if there's no sign of disease, especially if you're in a yard with a sign of disease, that means that the bacteria can be present. So you want to think about doing your treatments at the yard level and not just focusing on colonies where you see active signs of disease. There is a very strong seasonality with the signs and symptoms. So we see it a lot in Michigan. In June is the biggest peak, and then it kind of dwindles and hangs on over the summer, which is important for diagnoses. If it's later, like it's September, you're probably going to be looking at parasitic mite syndrome. It is really well characterized by being the most variable brood disease. So if you start to see that yellow, you start to see twisted, you start to see all different shapes and sizes, that would be one of the main things that makes me think about European Foulbrood. And then it's just going to just show up in different hives every year. Again, going back to that treatment at the yard level is kind of try to think about it as this operation-wide pathogen. So again, you can always ask questions in the future by looking at or going to pollinators.msu.edu and filling in the questions. Someone had asked if that's only Michigan specific, and not every extension uses that site. However, it's national. Even we'll take international questions as well. And so if there is something that is like it's useful to have a local person, they'll reassign it to someone local, but the bee health team isn't so huge. So it may come to me, it may come to somebody else, but we can always reassign it as well. Peter, I'm just going to feed you questions and answer some of them myself. So the first one, I'm pretty sure that Peter wrote in as a plant. And it says, do we know how many different strains of EFB there are? And then the second point is how many are resistant to antibiotics?
So this disease is severely understudied. Right now, they've identified, I think, 37 unique strains, and they mainly fall into 3 big genetic groupings. The 2 main ones we have here in the United States, that typical and atypical, are kind of the main broad genetic groups. As far as resistance, like I said, the resistance is fairly rare, but what they are finding with strains, which relates to resistance, is that it doesn't seem to depend on the strain type, the resistance, and they're not sure what mechanism. It's not a gene that's known to be associated with bacterial resistance, because most of these have been studied in human medicine. So they're actually not sure what's causing that different, that slightly higher resistance, but it's very rare, and they found it in strains from kind of each of the groupings. I think 4 have been identified so far. There was 4 in the Japanese study, and there's been 1 identified in Canada. So it's not very common, and again, we don't really know much about what that means at the colony level.
Yeah. And I think one of the things is just trying to, we're for the very first time looking at what strains are circulating in Michigan. So that is one of the things that I think, and then because we are doing full sequencing for a lot of them, I think we'll be able to get a lot of information on what those strain differences mean. Hopefully,
yeah, I can show the chart if you want.
It's a pretty chart. You should show it.
These are the strains we've isolated. This is like a subset of the strains we've isolated from Michigan. We took samples out in California, some down in Georgia. So trying to get a big sample pool. And this is the genetic relationship. And basically what you're looking at right here is 4 strains. So each of the colors is actually a different strain. And then all these different little lines in here, just how they're related. And you can see the red and yellow, those are in that atypical genetic cluster. And the little red asterisks, those are the ones that, from this grouping anyways, that were symptomatic with EFB when we sampled them. You can see a lot of these ones in the typical group didn't have symptoms, at least when we sampled them. So there's a lot more research to be done on, what's going on with these different strains. So.
And this is unpublished, so this is probably going to get cut from the recorded one. So you guys are lucky who stuck around to see it. So there are some questions about the other states that have the relationships with blueberries. And I don't have the data for those, but I know there are other states that are starting to look at this. And like I said, they've done a lot of work in British Columbia as well. But you definitely can get European Foulbrood without having any blueberries. One of the things that might be happening in Michigan and like other places is that, blueberries and all flowers bloom in response to usually degree days, which means that you have to have enough days above a certain temperature. So a lot of times people use like degree days base 50, and that's how many days where it's been or how much time it's been above 50 degrees. And when blueberries bloom, it's also the same time when lots of other things bloom. And so what happens is you do get this really big shift in colony dynamics because everybody goes out to forage. We also still have really cold nights. And so you have these changes in, maybe bees are no longer putting so much effort into nursing care. And so you do have these hive dynamic shifts that are at the same time, or these colony dynamics, and that could be part of it. What we do know is just having more food incoming, even though that's something people are like, oh, it'll clear up on a nectar flow. That is not our truth at all. There is a question about the different types of bees, like Italian, Russian, or Saskatraz. We have not looked at that. We did kind of mark queens at some of the operations, but I can tell you that in the colonies that we examined, it was a wide diversity of queens. So I don't know if there are strains that are more resistant. I do know that many, many strains are susceptible. All right. Oh, do you want to talk about the plasmid, what we've found in Michigan at all?
Oh, yeah. As far as what was the specific question about it?
They said, have you been able to sequence M. plutonis from the hives that did not show disease and the presence of the toxin carrying plasmid? So I think the M19.
Yeah. So, like I said, it's kind of tricky because the plasmid is really, it's the typical genetic cluster that depends on that plasmid for its virulence, as far as they know in the lab. What we're finding is a lot of these colonies, like half of them had both atypical and typical. And when you culture things for whole genome sequencing to get the DNA sequence, you're really just picking one to go with. So you're not sure if that really represents the whole picture of what was going on at the colony level. But we have done some screening for that. And the plasmid's definitely, I would say it's maybe in 3/4 of the whole genome stuff we got back. And when we screened using PCR, which is we're just looking for a chunk of that plasmid that's unique to that plasmid, we found all the samples that were in sick colonies had that plasmid present. They could be playing a pretty significant role, but there's a lot more research to be done to kind of tease out all those dynamics.
Mm-hmm. There's quite a few questions on freezing, and at least in terms of persistence and growth of the bacteria, we can freeze it for years and still have it grow, which is really unfortunate because, a nice freeze thaw cycle is a way to reduce pathogens. And it's not saying that it won't maybe reduce it on your equipment, but just freezing stuff alone will not kill the bacteria, unfortunately. So we can put it into a freezer and then plate it out years later and have it grow. There's a lovely question about, does Melissococcus plutonis naturally exist in the environment, soil or water? So can they pick it up and bring it back to the hive to start a bacterial infection in the colony? The short answer is I don't know, because I haven't found studies on it. There's one study that looks at nematodes. They found that the bacteria, that Melissococcus was living in these near these nematodes, but it really is severely understudied. However, there is a PhD student at Washington State that is starting to look at that. And so I'm just excited that, because there's so little research, it's great to have people going out there and seeing where it does exist. We don't have any evidence that it grows anywhere else. So it does seem to have to, if it's going to reproduce and grow and metabolize, that has to be in the bee. So Peter, there's a lot of questions on disinfection. Do you want to talk about what we know or what's been published on disinfecting equipment, and then maybe what we do for hive tools?
Yeah, so biosecurity has been a big it's big in the veterinary field, but not so much practice in beekeeping. Hopefully it gets better, but part of it's because we don't have very good recommendations. There's been a couple studies on what kills the bacteria, and unfortunately they're not very relevant. One of them was done in Japan on kind of a very specific disinfectant that I haven't been able to find anything similar over here. And then another study was done by that original scientist, the USDA scientist, like 100 years ago, and he was exposing it to different acids and then feeding it to colonies to see if it caused disease. So there's nothing actually that's testing stuff that I think we could commonly use. I saw some stuff was looking at asking about melting the wax down. Hopefully what we find is that this bacteria is pretty easy to kill, but the fact that it's persisting on all these surfaces to me suggests that there's probably a formation of some biofilm, and a lot of times this can protect against disinfectants, especially if it's in these porous surfaces. So I think it's something that we kind of don't have a lot to go on right now. I don't know if you have anything to add, Megan.
No, I mean, just that I'm thrilled that we have funding to look into it this year.
Yeah.
So the question, if it can be melted down, is there are no honeybee diseases that affect humans. So you can make chapstick out of EFB and go crazy for it.
They have found Melissococcus. It's related to it, but not specifically in the human gut. So where this bacteria came from evolutionarily is the biggest question to me. So I think a lot of that is environmental. Is it hanging out or a reservoir somewhere else is kind of an interesting question.
Yeah. All right. So Robin asks, if you do not treat and let it play out, will the hive build up its own resistance and less likely to reinfect, or just the opposite? If it gets it, will the hive continue to be susceptible? That is a very excellent question that we do not know. And part of it is just the difficulty of queen dynamics and bee dynamics. The bees who are exposed to it are going to be dying very soon. And then if the queen gets it, whether or not she's able to pass something down, I've talked to people that, maybe that's why we see 1 year it's healthy, 1 year it's sick, and 1 year it's healthy, 1 year it's sick. There's definitely a lot of dynamics, but I would say for as well studied as bees are, we're still learning about their basic immune system. There's a lot that we still have to learn about. All right, so there's some questions about using the VITA test rather than interpret the sides of melted brood to find out if you have AFB. So we do like the VITA test. We've not done a validation study for the AFB one. Another thing though is we are posting very, very soon a online module that is on diagnostics. So this was part of our veterinary training, but we're making it available to beekeepers because everyone should be able to have access to diagnostic tools. And Peter's actually taken some lovely media for that. But there are quite a few in-field tests for American Foulbrood, especially, that can help you roll that out. And then I don't know, there's a question if those test kits can be used after expiration. That would be something I would ask the company. I have used them after expiration and have gotten positives. I would be more worried, though, about a false negative in that case. So if you got a negative with an expired test, I don't know that I would really trust it. Let's see. Did you talk about the hive tools, about how we clean them?
Oh, yeah. So usually our protocol for cleaning hive tools is a lot of Comet, and those green scrubber pads are great. Basically, the main thing is getting all the physical material off of it. So getting all the propolis off the tool is kind of like first goal. And then as far as disinfecting, soaking it in bleach afterwards. I usually use like a 10% bleach solution is usually, you can go more concentrated. Soak it for a little bit and then just air dry. You can throw it in a pressure cooker if you want to be extra safe. But yeah, that's our general hive tool cleaning method. I don't know if you have anything to add as far as.
I think the big thing is getting everything off of it.
Yeah.
So a lot of the, scraping with the other tools, the way that I like to do it in my own operation that we do it at MSU is just to have multiple tools, so you can kind of build up a bunch that need to be washed and then wash them at the same time. There's a question about, does scorching the boxes help? When you scorch with like a blowtorch or a flame weeder, one of the biggest things about that is it's very variable. because the bacteria need heat at a certain amount of time. And so when you're scorching, because the wood's so porous and that flame is so variable, it definitely will reduce the amount of pathogen. And this is just for any pathogen, but it won't necessarily get rid of it. If you can do prolonged heat consistently over a very long time, so for example, an autoclave, dipping in hot wax, Australia has a lot of resources, but that's one of the techniques that I use. So that would be hot wax at 130 degrees Celsius for over 10 minutes, I believe. And then I also use a dry heat oven, but you can also use a steam melter. So I do 500 degrees Fahrenheit for 5 hours on frames, and that should kill quite a bit of it. You really want consistent, prolonged heat. I can see people trickling off. We'll just keep it going until everyone's gone or we run out of questions. If there's a sick hive that's not gaining strength fast enough to outcompete the robbing pressure, if you cannot move the hive, should you euthanize the colony and how? It's so complicated on many levels. Exactly, Gray. So we've done a couple of things. We had exactly this situation last year, and we did it as a case study for the veterinary students where there was one itty bitty colony and one pretty healthy colony. And it was just those two in that yard. And they'd been side by side all summer. And this was getting late in the season, so we wouldn't be able to really save that little colony. What we decided to do in that case is we shook the bees out of the little colony and took that equipment home, got rid of the frames, and cleaned out the boxes. The idea is that even though it probably is in those adult bees, those adult that are going to go into that other hive, those adult bees were already, since it was two hives right next to each other, they were probably already going into each other's hives. You can just euthanize the colony as well. I'm actually on the committee for writing the AVMA regulations of how we recommend euthanasia or depopulation for a honeybee colony. There's not a great method. You can use an insecticide spray. Is quite quick. There are some studies that have published that if you use a permethrin, which is the natural version of the pyrethroid version of insecticide spray, you can actually reuse the comb after that because it breaks down so quickly. The standard that they've been using in Australia is gasoline. So you're using the fumes to kill off the bees. And there are recommendations for that, but it is something you want to be very, very careful. Or the other thing that that I've recommended is shaking the bees into like a dishwasher tub of alcohol. The ones where you pour things over the hive doesn't go quickly enough. Jeannie asks or says, I read that earwigs may serve as a vector for EFB. Is this correct? And are there other vectors? I do not know at all about earwigs, and I haven't read that, but we did have a beekeeper recommendation to look at hive beetles. We did try to collect. I don't think we were able to test any of them.
Yeah, we've actually got quite a few from some pretty sick hives. We haven't tested them yet, though.
Okay.
So that's pending.
I mean, earwigs you see a lot in hives. The reason we looked at hive beetles is because they fly from one to the other. Let's see. Do you see any that look like we haven't covered yet?
One down at the bottom asking about irradiation and fumigation.
So, yeah, and I've actually, in the last 3 days, I think I've given my talk on cleaning hive frames 3 times. So I wasn't sure if we had talked about that yet. In Michigan, we do have an irradiation facility that is close by that used to have beekeeper days. I haven't checked to see if they still do. But you could bring in your equipment. That is something that is possible. That would be a case in which they would sterilize it. One thing that is really hard, though, is that when you have a honeybee colony, they're not sterile themselves. So if you sterilize your equipment, they're going to be dirty in like 6 seconds afterwards. Acetic acid fumigation is on our list of things to study when we look at disinfectants, but that's something that I've not been able to find data for Melissococcus yet. All right. So I think we're quite a bit slowed down, but like I said, we'll happily take many more questions through the pollinators at msu.edu Ask extension, and hopefully we can do another one of these next year and give you way more positive updates, and we'll have everything totally solved, she says. So I just want to thank you so profoundly for taking time out of your day to listen to us and learn more about your bees. And hopefully we'll see each other at a conference at some time soon. Thanks, everyone.
Thanks, everyone.